Acha, so in the last class we were doing section number 1, which was cellular organelles, and we were done with almost all the organelles except these three: cellulose cell wall, permanent vacuole and tonoplast, and your plasma desmata, which we'll be doing today. And in tomorrow class we'll start microscopic section. So let's see. Before we move towards plant cell wall, first let's look at some high yield questions for your paper 1. So mostly in your paper 1, which is your MCQs, they ask for structures with double membrane or structures with no membrane and structures which contain cisternae. Cisternae क्यों? Because they know, CI wale, they know कि students tend to mess up cisternae with cristae. So you should know cisternae कहाँ पर होता है and cristae कहाँ पर होता है. So let's see. Double membrane is found in your nucleus, inner and outer membrane with nuclear pore. It's found in your nucleus, in your chloroplast, and in your mitochondria, inner and outer membrane, but no pores. And it has a intermembrane space, which we did yesterday, mitochondria. So these three structures have double membrane, where no membrane is seen in your ribosomes only. सिर्फ ribosomes ऐसे structures होते हैं जिनके पास कोई भी membrane नहीं होती, whereas your lysosomes they have a single membrane. So ribosomes are the one with no membrane. Ribosomes. Here you have cisternae. Cisternae is found in your smooth endoplasmic reticulum, your rough endoplasmic reticulum, and your Golgi body. तो cisternae का shape किस तरह होता है? Let me show you. कुछ इस तरह होता है. या फिर if you're talking about your Golgi body, तो कुछ इस तरह होता है. Whereas cristae, which is different from cisternae, cristae is found in your mitochondrial chloroplast. And it is basically the folding of your inner membrane. जब आपकी inner membrane fold करती है इस तरह करके, it forms cristae. इन दोनों में आपको differentiate करना आना चाहिए. Something like this. This is the structure of your mitochondria, जिसमें जो आपकी inner membrane होती है, it is folded to form cristae. Now for today's class, we'll be studying plant wall, plant cell wall. Start करते हैं. Let's see. Give me a minute. So your plant cell wall, it is made up of polysaccharide, which is known as cellulose. कौन से polysaccharide से बनी होती है? That is cellulose. And your cellulose, it exists as fibers. किस तरह होती है arrangement? Something like this. These fibers are arranged in a crisscross manner. This is what you call a crisscross manner. And these are your cellulose fibers. Now due to this crisscross manner, आपके जो cellulose है, cellulose cell wall, इसमें pores बन जाते हैं. Which is why your plant cell wall is permeable in nature. It allows the movement of substances in and out through it. Now, your cellulose fiber, something special about it is it has a high tensile strength. It is very strong, tensile strength, and it basically prevents your cell from bursting. जब भी आप इस सेल को अपने high water potential solution में रखोगे, your cell won't burst due to your cellulose fibers. Now, why is the cellulose fiber permeable in nature? Why is your plant cell permeable in nature? Due to the presence of pores, and these pores are made by the crisscross arrangement of your cellulose fiber. So basically these pores are accounting for the permeable nature of your cell wall. Permeable nature gives permeable nature to the cell wall. They won't ask you to draw the structure of your cell wall, तो इसकी टेंशन की कोई बात नहीं है. Now what is the role of your plant cell wall in basically your plants? So very first example, your very first role is it provides mechanical strength to your plant. And how is that provided? Due to the presence of your cellulose fibers. Mechanical strength due to the presence of your cellulose fibers. And वो क्यों होता है? क्योंकि your cellulose fibers, they have a high tensile strength. They're very strong in nature. The second function of your cell wall is it protects the protoplasm. Protoplasm is basically the thing inside your cell wall. आपका टोनोप्लास्ट, आपका वैक्यूल, न्यूक्लियस, साइटोप्लाज्म, ये सारी चीजें दे आर पार्ट ऑफ योर प्रोटोप्लाज्म. तो जितनी भी चीजें होती हैं अंदर, दे आर प्रोटेक्टेड बाय द सेल वॉल. किस चीज से प्रोटेक्ट कर रहे होते हैं? फ्रॉम इन्वेडिंग माइक्रो ऑर्गेनिज्म. द पोर्स इन योर सेल वॉल आर नॉट टू लार्ज टू अलाउ बैक्टीरिया या फिर वायरसेस टू एंटर योर प्लांट cell. So it provides mechanical strength, and it protects your plant cell from invading microorganisms. The third function is it maintains your plant cell shape, which is why आपके जो plants हैं, जब भी आप इनका electron micrograph देखोगे, आपको एक proper arrangement में नजर आएंगे, regular arrangement में नजर आएंगे. Whereas your plant cell, animal cell, they have an irregular arrangement due to the absence of your cell wall. So they provide या फिर they maintain plant cell shape, plant maintain shape. The very last function is your plant cell wall. It may serve as a reservoir of food. Reservoir of food. These are the four functions of your plant cell wall which you should know. Tiny सा ये topic था आपका on your plant cell wall. And I've written this pointers down below. So एक बार go through कर लेते हैं. Plant cell wall is made up of polysaccharide, cellulose. We'll study the structure of cellulose in your chapter number 2, biological molecules. कि किस तरह बना होता है and कौन से glucose molecule से बना होता है ये. So basically it is made up of beta glucose. किस तरह बना होता है? We'll study in next chapter. Now your cellulose exists as cellulose fibers. ये रहा आपके cellulose fibers which are arranged in a crisscross manner. And I just told you इनके पास high tensile strength होती है, which is why it is able to provide mechanical strength to your plant. These fibers are arranged in a crisscross manner within the cell wall, leaving behind large pores which account for the completely permeable nature of your cell wall. इसके through water भी आ सकता है, इसके through food भी आ सकते हैं, molecules भी आ सकते हैं, ions भी आ सकते हैं, but इसके through bacteria and viruses नहीं enter कर सकते क्योंकि उनका साइज काफी ज्यादा large होता है. And compared to that, आपके जो cell wall में pores हैं थोड़े से small होते हैं, which is why protection मिल जाती है एक आपको protoplasm को. Now outline the role of cell wall in plants. They can ask you this question in CI. So आप क्या क्या लिखोगे pointers? Cell wall provide mechanical strength due to the presence of cellulose fiber. So this is point number 1. Cell wall protects the protoplasm from invading microorganisms. Why is that so? The pores in the cell wall are not too large to allow bacteria or viruses to enter. Here, cell wall maintains the plant cell shape. And lastly, cell wall may also serve as a reservoir of food. After this, we need to study large permanent vacuole and tonoplast. So let's see. किस तरह होता है? This is your plant cell. Basics है आपका plant cell. Let's label the structures जो हमें पता है पहले से. This is your cell wall. फिर अंदर वाली which is selectively permeable वो होती है आपकी cell membrane. Cell wall is completely permeable whereas cell membrane it is selectively permeable. कुछ चीज़ों को allow करती है and कुछ चीज़ों को allow नहीं करती. Here you have vacuole, large permanent central vacuole. Whereas in animal cell काफी छोटी सी vacuoles आपको नजर आती हैं. But in plant cell बिल्कुल opposite होता है. So this is your um vacuole तो है. Let's see. एक minute. The membrane of your vacuole is known as tonoplast. अच्छा, always remember whenever you see a hollow structure कुछ इस तरह करके, ये hollow नहीं होता actually में, there is some type of fluid present within it. तो यहाँ पे भी ऐसे ही हो रहा है. The fluid within your vacuole is known as cell sap. Cell sap. तो ये दोनों बनाते हैं आपके vacuole. Cell sap is the content within your vacuole and tonoplast is basically the membrane of your vacuole. Here, surrounding your tonoplast, you have chlorophyll. ये रहा आपके green color के chlorophyll. Chlorophyll. And ये edge पर क्यों होते हैं? To absorb maximum light for the process of photosynthesis. So this is a basic structure of your plant cell. इसमें we need to study the structure of your vacuole. So you should remember कि आपके plant में किस तरह की vacuole होती है. It has a large vacuole and it has a large permanent vacuole. The content of your vacuole are known as the cell sap, and your cell sap is enclosed within the membrane known as tonoplast. So the membrane covering your vacuole is known as tonoplast. Now this tonoplast, like your cell membrane, it is selectively permeable. It controls the substances that enter and leave your cell sap. कुछ कुछ चीज़ों को आने देती है, कुछ कुछ चीज़ों को जाने देती है, which is why we call it selectively permeable. So this is selectively permeable like your cell membrane. Permeable. And again, they won't ask you to draw the structure of tonoplast in your exam. Acha. Compared to animal cell, your animal cells have very small and temporary vacuole. छोटे छोटे से आपको एनिमल सेल में नजर आएंगे vacuole, and these are temporary ones, permanent नहीं होते. They get destroyed at some time, whereas in your plant cell you have a large permanent vacuole. तो ये थोड़ा सा आपका टॉपिक था on vacuole. Now what is the role of this permanent vacuole in plant cell? पहला तो is related to your cell sap. Let's see. आपका जो cell sap होता है, it contains a number of different type of material that can be beneficial for your cell. The cell sap contains water, it has sugar, it has amino acid, it has minerals, and it has waste material as well. Here, your vacuole, it maintains the turgidity of your cell, and it basically provides support and shape to your plant cell. Last benefit is आपका vacuole इतना बड़ा होता है बीच में कि it starts to push the chloroplast towards the edge of the cell. जिसकी वजह से your chloroplasts are able to obtain या absorb maximum light, and this light is used in the process of photosynthesis. So ये the third benefit for your vacuole. Let's see the pointers. I've mentioned these down below as well. Plant cell possess a large central vacuole. Animal cell may possess small temporary vacuoles but not a large permanent one. The content of the vacuole are termed as cell sap. क्या बोलते हैं इसे? Cell sap. Cell sap is enclosed within a membrane known as tonoplast. Tonoplast is a selectively permeable membrane which controls what enters and leaves the cell sap. तो बिल्कुल आपके cell membrane की तरह it is a selectively permeable membrane. Role of a permanent vacuole in plant cell: it stores cell sap which contains water, sugar, amino acid, mineral ion and waste material. Vacuolar turgidity provides support and shape to the plant cell. Vacuolar turgidity also pushes the chloroplast towards the edge of the cell to maximize light absorption for the process of photosynthesis. Photosynthesis. अगर कहीं भी कोई difficulty आ रही है, you can open your mic and let me know. So yeah, I'll re-explain the concept to you. Achha, the last organelle that we need to do is plasmodesmata. This is how your plasmodesmata looks like. It is between your two adjacent plant cells. एक plant cell ये रहा आपका, and this is your second one plant cell. ये दो plant cell के बीच में आपको नजर आता है. किससे बना हुआ होता है? It is basically made up of your cytoplasmic strand. And क्यों होता है ये? Let's say इस plant के पास ज़्यादा sugar है and this plant is lacking sugar in it. तो अगर इस प्लांट को शुगर चाहिए, इट कैन डायरेक्टली टेक इट थ्रू द प्लाज्माडेस्माटा. इसे मेम्ब्रेन के थ्रू पास करना नहीं पड़ेगा. अगर मेम्ब्रेन के थ्रू पास करेगा तो बहुत ज़्यादा टाइम वेस्ट हो जाएगा. हो सकता है आपका सेल तब तक डाई कर जाए ड्यू टू लैक ऑफ शुगर, या सर्टेन प्रोसेसेस इसके अंदर नहीं हो पा रहे हो. तो इंस्टेड ऑफ टेकिंग टू मच टाइम एंड पासिंग थ्रू द सेल मेम्ब्रेन, it can directly pass through your plasmadesmata. Basically, it allows rapid communication between your adjacent plant cells, ताकि आपकी सब्सटेंसेस को सेल मेम्ब्रेन के थ्रू पास ना करना पड़े. फिर it allows this movement of your water. It allows the movement of your sugars, amino acid, proteins, hormones. Basically, everything can be transported between your adjacent plant cell. So this was also a very chota sa topic on your plasmodesmata. Only two pointers. This is your plasmo. Plasmodesma. Singular is plasmodesma, plural is plasmodesmata. आई होप आपको इजी लग रहे हो, इतने डिफिकल्ट टॉपिक नहीं है, छोटे छोटे से टॉपिक हैं. A plasmodesmata is made up of cytoplasmic strands which run transversely across the walls of a plant cell. And two functions है इसके छोटे से. It transports water, sugar, amino acid, hormones, etc. between an adjacent plant cell, and it allows rapid communication between the adjacent plant cell क्योंकि आपके substances को pass नहीं करना पड़ता through the cell membrane. It directly passes through your plasmodesmata. Now, before we conclude the class, let's look at the relative size of cell structure. This is again a P1 paper 1 pointer. पेपर वन में आपसे पूछा जाता है कि कौन से सेल स्ट्रक्चर का साइज सबसे बड़ा होगा, कौन से सेल स्ट्रक्चर का साइज सबसे सबसे छोटा होगा. टू मेक दिस इजियर फॉर यू, देयर इज अ ट्रिक यू कैन लर्न डायरेक्टली. एंड वो ट्रिक क्या है? नोबडी आई थिंक आपने सुने भी होगी, कैन मेक नूडल्स लाइक शेफ रैमजे. शेफ रैमजे तो सबको पता है कौन है, गॉर्डन रैमजे, काफ़ी फेमस शेफ हैं, एंड उन्हीं से रिलेटेड ये ट्रिक है. तो व्हाट इज द फुल फॉर्म ऑफ दिस, जल्दी से देख लेते हैं. N stands for nucleus. So nucleus का साइज सबसे ज्यादा बड़ा है, nucleus. फिर C stands for chloroplast. M stands for mitochondria, mitochondria. And again stands for nucleolus, nucleolus which is present within your nucleus. किस तरह होता है? ये आपकी nucleus and dark grainy substance जो आपको नजर आ रहा होता है, that is your nucleolus. So this is your nucleolus. L stands for lysosome. Lysosomes contain your hydrolytic enzymes. C stands for centriole. It helps in the process of cell division. R stands for ribosome, which is involved in protein synthesis. जल्दी जल्दी हमने थोड़ा सा recap भी कर लिया. So the largest organelle is your nucleus. This is your largest organelle. And the smallest organelle is your ribosome. आपको easy सी एक ट्रैक मिल गई. And you can easily and quickly solve your paper 1 के MCQs. And we'll apply this जब हम practice करेंगे past paper. I think Monday को ही करेंगे. Also, I need to share the past paper booklet with you, so I'll share it today, right after the class. Now we're done with this, with eukaryotic cell structures and cell organelles. Now we need to move towards the structure of your prokaryote. Prokaryote is what? Your bacteria. Bacteria क्या होता है आपका? Prokaryote होता है. So let's first try to identify the cellular organelles in your prokaryote. Now, the organelles in your prokaryote are divided into two classifications. एक ऐसे ऑर्गेनिज्म हैं जो हर वक्त प्रेजेंट होते हैं, हर बैक्टीरियल सेल में प्रेजेंट होते हैं, बट देर आर सम स्पेशलाइज्ड सेल ऑर्गेनिज्म या सेल स्ट्रक्चर्स विच आर प्रेजेंट इन सम बैक्टीरिया. आपको वो हर बैक्टीरिया में नजर नहीं आएंगे, तो मैं दोनों की क्लासिफिकेशन कर देती हूँ इन द इजिएस्ट वे पॉसिबल. फर्स्ट वी आर गोइंग टू लुक एट सेल ऑर्गेनिज्म दैट आर प्रेजेंट इन ऑल टाइप ऑफ बैक्टीरियल सेल्स. so the cell organelles which are always present वो हम यहाँ पे लिखेंगे ताकि आपके लिए easy हो जाए when you look back at it. And the cell organelles which are present in some of the bacteria may be present, किसी किसी के पास होते हैं ये, वो हम यहाँ पे list down करेंगे and will label this diagram as well. तो सबसे पहले, like animal cell, bacterial cell also have a cell wall, but इनके cell wall is made up of another type of substance. वो substance क्या होता है? That is peptidoglycan. So the cell wall of bacteria, your prokaryote, is made up of cell wall peptidoglycan. Made up of peptidoglycan. सबसे आउटर वाली लेयर इज योर सेल वॉल. फिर इनर वाली क्या होती है? सेल मेम्ब्रेन होती है, एग्जैक्टली एज योर एनिमल सेल या यूकैरियोटिक सेल. So this is your cell membrane. फिर इफ यू लुक एट दिस, आपके पास सर्कुलर डीएनए होता है. ये रहा. This A type thing is your circular DNA. Circular DNA. The next thing, your circular DNA is surrounded by a membrane, a discontinuous type membrane, and this is known as nucleoid. Nucleoid. Here you have ribosome. कौन सा ribosome? Your 70S ribosome. 70S ribosomes आपको तीन चीजों में नजर आते हैं. In your mitochondria, in your chloroplast, and in your bacterial cell. Why are they present in mitochondria and bacteria? Sorry, chloroplast. वो हम study करेंगे in your endosymbiotic theory. So these are your 70S ribosomes. फिर this fluid is your cytoplasm, same as the eukaryotic cells. इसमें कोई difference नहीं है. This is the cytoplasm. ये हर bacterial cell में आपको नजर आएगा. So there were one, two, three, four, five, six structures that are always present in any bacterial cell. Now let's look at the four structures which are present in some bacterial cell. हर bacterial cell में नजर नहीं आएगा आपको ये, specialized bacterial cells में नजर आएगा. So the very first obvious one जो हमें नजर आ रहा है, पहले हम वही label कर लेते हैं. This one. This is a flagella. Flagella या फिर flagellum. This helps your bacteria to move from one place to another. Here you have the slimy capsule. Slimy capsule. सबका function हम पढ़ेंगे one by one. This is your slimy capsule. Here you have your pili. This sharp, spiny thing. This is your pili. Pili. And this turquoise type thing, थोड़ी सी जो बड़ी सी नजर आ रही है, bigger than your ribosome, ये होते हैं आपके plasmids. And ये सबसे important feature होते हैं आपके बैक्टीरिया के, because ये आपके genetic technology में help कर रहे होते हैं. किस तरह? We'll see in a while. Now, if you remember our very first lecture, जब हम discuss कर रहे थे type of nucleus, तब हमने डिस्कस किया था देयर आर टू टाइप्स ऑफ न्यूक्लियस. एक ट्रू होती है, एक फेक होती है. ट्रू न्यूक्लियस वो होती है विच आर सराउंडेड बाय न्यूक्लियर मेम्ब्रेन एंड दे फाउंड इन योर यूकैरियोटिक सेल्स. whereas fake nucleus, apparently fake nucleus, they are not surrounded by a nuclear membrane and इनका जो डीएनए होता है that is circular DNA. whereas true nucleus में यहाँ पे मैं मेंशन कर देती हूँ, it has linear DNA. and fake nucleus में you have circular DNA. तो ये भी एक important classification है, the shape of the DNA. So your prokaryotic cells, they have fake DNA, circular DNA, which is devoid. They're devoid of a true nucleus. Let's go back here. Now the region of the cytoplasm where the circular DNA lies is known as your nucleoid. And your circular DNA is not naked, is naked, sorry. It is not bound by histone proteins. आपको जो normal eukaryotic cells में DNA था, वो कुछ इस तरह था. चाहिए? Let me draw the shape for you. कुछ इस तरह था. And ये जो red things है, these are your histone proteins. And बाकी ये जो strand नजर आ रहा है आपको, this is your DNA. But this was in your eukaryotic cell. Whereas in your prokaryotic cell, जो आपको bacterial cell है, आपके पास इस तरह करके circular DNA होता है, double stranded circular DNA. And इसमें आपको histone proteins नजर नहीं आते. So your DNA is basically naked and it is not bound by histone protein. The size of your prokaryote, it ranges from 2 to, sorry, 0.5 to 5 micrometer. This is the basic range of your prokaryote. ये आपको याद होनी चाहिए. This is asked in paper 1. And if you look closely, if you observe the diagram, आपको कोई भी membrane bound organelle नजर नहीं आएगा. कौन से membrane bound organelle? Like your mitochondria, your chloroplast, your nucleus, your RER, SER, Golgi, lysosome. कोई भी आपको एक भी membrane bound structure नजर नहीं आएगा. And the cell wall of your prokaryote, it is made up of peptidoglycan. Peptidoglycan is basically a modified polysaccharide, which we'll study in detail in a bit. So I've written these pointers as well. You can go through them once the class is over. Same pointers यहाँ पे भी लिखे हुए which I just discussed. Let's discuss the cell wall of your bacterial cell. Now, it is made up of peptidoglycan, which I just told you is a modified polysaccharide. But ये किस चीज़ का polysaccharide है? It is basically the polysaccharide, a polymer of a sugar and amino acid, which is why it is also known as murin. इसको क्या बोलते हैं? It is also known as murin. Like, give me a minute, I'm coming. Let's continue. Okay, so we were discussing cell wall. It is made up of peptidoglycan, which is a modified polysaccharide, and it is a polymer of your sugar and amino acid, which is why it is also known as murin. Now this cell wall, like your normal cell wall, ये भी आपको mechanical strength provide कर रही होगी to your bacterial cell, and it will prevent your bacterial cell from undergoing lysis. Lysis क्या होता है? Cell death. जब आपकी cell की death हो जाती है, that is known as lysis. So basically it prevents the cell from dying due to turgor pressure. Turgor pressure क्या होता है? Pressure due to the water. Turgor pressure of the intracellular content. तो अगर water enter होगा आपके cell के अंदर बहुत ज़्यादा क्वांटिटी में, it will swell but it would not burst. Burst होने से prevent करेगी, which is why it is providing mechanical strength to your bacterial cell. Now the cell wall, it also protects the bacteria from attack by the immune cell. कौन से immune cells? Your phagocytosis, your bacteriophages. Bacteriophages are basically ऐसे viruses that attack your bacteria. So we'll study this in our last chapter, which is immunity. So एक protection भी दे रही है आपके cell को. So let's look at the pointer on the next page. It is made up of peptidoglycan, which is a polymer of sugar and amino acid. Peptidoglycan is also termed as murin. Cell wall provides mechanical strength to the bacterial cell, preventing it from undergoing lysis due to turgor pressure of the intracellular content. Cell wall also protects the bacteria from attack by immune cell. Immune cell कौन से हो सकते हैं? Your phagocytes. For example, your macrophages, monocytes. Phagocytes include your monocytes and your neutrophils. अभी ये थोड़ा सा कॉन्सेप्ट समझना मुश्किल है इसलिए मैंने लिखा ही नहीं डायरेक्ट. So your phagocytes are basically your monocytes and your neutrophils, which we'll study in last chapter. Neutrophils, neutrophils or viruses such as bacteriophages. Bacteriophages उन viruses को कहते हैं which attack your bacteria. Now, the circular DNA. Circular DNA is naked. Why is that so? Because it is not bounded by your histone protein. Normally, histone proteins इस तरह होते हैं कुछ. And it is found in your linear DNA. But here structure is absent in your bacterial cell. And your circular DNA contains bacterial gene obviously which code for bacterial protein. ये तो basic सी बात है. Now you have 70 ribosomes exactly the same as the ones in your eukaryotic cell. एक large subunit होता है and एक small subunit होता है and each subunit contains protein and RNA. Proteins plus ribosomal RNA. So the same thing is going on नीचे वाले subunit में भी. And they're involved in protein synthesis via translation. बिल्कुल exactly same as your eukaryotic cell. The cytoplasm, it contains soluble enzymes, 70S ribosomes, and circular DNA. These enzymes catalyze hydrolytic reactions within the cytoplasm. The slimy capsule, ये थोड़ा सा नया कॉन्सेप्ट है. ये आपका slimy capsule, बिल्कुल एक कैप्सूल बन रही है यहाँ पे बाहर, external to your cell wall of the bacterial cell. Basically, it is an additional layer to your cell wall. Cell wall के ऊपर होती है ये, and it is not present in all bacteria. कुछ कुछ bacteria में present होती है. So what is the function of this? दो function है. एक तो ये phagocyte से protect करेगा. जब आपके immune cells attack करने आएंगे तो ये bacteria resistant हो जाएगा to the attack of your phagocytosis. फिर it will prevent the bacteria from drying out. Let's say bacteria बहुत dry environment में है and temperature बहुत ज़्यादा है उस environment का. So it will prevent your bacteria from drying out. एंड बिल्कुल एग्जैक्टली यही यहाँ पे लिखा हुआ है. इट इज एन एडिशनल लेयर एक्सटर्नल टू द सेल वॉल. It is not present in all bacteria. Slimy capsule preventing the bacteria from drying out and protecting it from phagocytes. Phagocytes दो ही होते हैं, monocytes and neutrophils. Monocytes plus neutrophils. Flagellum. Flagellum is derived from microtubules. Microtubules we studied, they were a part of your cytoskeleton. तो माइक्रोट्यूब्यूल से आपकी फ्लैजिलम भी बनती है एंड इट इज नॉट प्रेजेंट इन ऑल बैक्टीरिया. स्पेशलाइज स्ट्रक्चर है ये. इफ प्रेजेंट इट असिस्ट इन योर बैक्टीरियल मोटिलिटी. किस तरह असिस्ट करता है? इट कॉन्ट्रैक्ट्स एंड रिलैक्सेस, जिसकी वजह से आपका बैक्टीरिया एक प्लेस से दूसरी जगह जाता है आराम से. बेसिकली स्विमिंग में हेल्प करता है आपके बैक्टीरिया को. लास्टली द मोस्ट इंपॉर्टेंट थिंग इज योर प्लाज्मिड. प्लाज्मिड बेसिकली आपकी सर्कुलर रिंग्स ऑफ डीएनए होती हैं. Basically, ring itself is made up of DNA and it is not present in bacteria. Again, specialized structure है ये. Now, plasmid का function क्या होता है? Gene technology में help करती है ये. Why is that so? Because it contains antibiotic resistance gene. Antibiotics बनाने में help करती है. So it will provide you protection against a particular antibiotic. And यहाँ पे लिखा हुआ भी है, plasmids are also used as vectors in your genetic engineering, which we'll study in A2 inshallah. Now the last thing that we need to study was a pili. Pili पे they can ask you a question. Pili क्या था? Let's go back. ये spiky structure. I think you can predict कि किस चीज़ के लिए यूज हो रहा है. एक तो ये अटैचमेंट के लिए यूज होगा एंड द सेकंड वन विल सी here a question. Briefly describe the role of pili in bacteria. So there are two types of pili. दोनों डिस्कस करते हैं हम. One of them is known as attachment pili, and as the name suggests, it will help your bacteria to attach to a particular surface, also known as fimbriae. And the second one is your sex pili. It basically enables conjugation between your bacteria. It allows the two bacteria to reproduce. So enable conjugation between bacteria. किस तरह enable करेगा? Let's say this is your bacteria, अजीब सा bacteria, and ये रहा आपका दूसरा bacteria, and ये रहे इनके पिलाई, the red thing. ये पिलाई से connect करेंगे कुछ इस तरह. दोनों के पिलाई कनेक्ट करेंगे and it will form a pathway for the transfer of substances from one bacteria to another. तो यहाँ पे जो भी चीज़ें होंगी, let's say कुछ स्पेसिफिक प्रोटीन्स हैं, कुछ भी हैं, दे विल ट्रांसफर फ्रॉम हेयर, यहाँ से पास करेंगी थ्रू द पिलाई एंड इसमें आके एंटर कर जाएंगी. So this process is known as conjugation. These were the two functions, the role of your pili in bacteria. Last thing that we need to see before we end our class is viruses. बहुत सिंपल सा टॉपिक है. You study this mostly in your MD-CAT, जिससे पूरा एक अलग से चैप्टर है and it is very long and difficult. But यहाँ पे in your A levels it is very easy, छोटा सा टॉपिक है. So viruses range in diameter from 20 to 300 nanometer. Self-explanatory pointer है. There's no need to explain in this. अच्छा. The larger sizes of your eukaryotes. Eukaryotes. फिर you have the prokaryotes. And सबसे small होते हैं आपके viruses size में. And this is a P1 pointer. The size is your P1 pointer. Usually आपके paper 1 में पूछते हैं. They do not ask you this in paper 2. इस तरह आपका size increase हो रहा है. So you can learn this a day before your paper 1. Achha, they are smaller than your eukaryotic and prokaryotic cells. And viruses are not yet considered as living things. Why is that so? जब आपके वायरस होस्ट सेल से बाहर होते हैं, ये बिल्कुल बेकार होते हैं। द एग्जिस्ट इज नॉन लिविंग थिंग, कुछ भी नहीं कर रहे होते, इनकी एक्टिविटी बिल्कुल डेड होती है। जैसे ही होस्ट सेल के अंदर एंटर करते हैं टू सम प्रोसेसेस, ये फौरन से एक्टिवेट हो जाते हैं। जब ये बाहर होते हैं दे बिहेव लाइक नॉन लिविंग थिंग, फिर जब ये एंटर करते हैं बिहेव लाइक लिविंग थिंग। वाई इज दैट सो? जो वायरस की प्रोटीन मशीनरी होती है, प्रोटीन मशीनरी इज अनदर नेम फॉर योर राइबोसोम। उनको यूज करके ये अपने प्रोटीन्स बनाते हैं एंड उसकी वजह से ये रेप्लिकेट करते हैं। So they start to replicate with the help of the protein machinery of your host cell. जल्दी जल्दी नंबर बनाते हैं एंड इतना ज़्यादा इंक्रीज कर जाते हैं क्वांटिटी में कि इवेंचुअली हमारी सेल वॉल नहीं होती, जिसकी वजह से सेल बर्स्ट हो जाता है। और जब सेल बर्स्ट होता है कुछ इस तरह, तो ये सारा बैक्टीरिया, सारा वायरस निकल जाता है एंड स्टार्ट टू स्प्रेड इन योर बॉडी। तो ये पूरी कहानी चल रही थी, विच इज़ वाई दे आर नॉट येट क्लासिफाइड एज लिविंग थिंग, क्योंकि जब ये बाहर होते हैं बिल्कुल बेकार होते हैं, कोई काम नहीं होता इनका। लेट मी इरेज दिस। सिंपल वायरस का स्ट्रक्चर किस तरह होता है? दो चीज़ें, सिर्फ 2 पॉइंटर्स, एंड दैट इज इट इज मेड अप ऑफ अ प्रोटीन कोड। इस प्रोटीन कोड का नाम होता है कैप्सिड। कैप्सिड का फंक्शनल यूनिट क्या होता है? कैप्सोमेयर। कैप्सिड इज मेड अप ऑफ कैप्सोमेयर and then it has nucleic acid. Now this nucleic acid can either be DNA और RNA, कोई एक होगा इन दोनों में से एट अ टाइम, दोनों एक साथ नहीं हो सकते। So it can either be a DNA or RNA, and it can either be a single stranded DNA या फिर double stranded DNA. In the same way it can either be a single stranded RNA या फिर double stranded RNA. This one. And I've written this here as well. A simple virus is made up of nucleic acid, DNA or RNA, and a protein code capsid. So let's label this here. ये रहा आपका कैप्सिड। एंड पेपर में भी बस आपसे ये दो टू स्ट्रक्चर्स के बारे में ही पूछेंगे। द नोट्स आर वेरी सीआई ओरिएंटेड एंड मार्किंग स्कीम से ही लिए गए हैं पॉइंटर्स। So it's made up of capsomer protein and it has a single or double stranded DNA or RNA. Now the nucleic acid contains viral genes. जैसे nucleic acid of animal cell had animal genes, when nucleic acid of bacterial cell had bacterial genes. In the same way, nucleic acid of virus has viral gene, which will code for viral protein. The capsid protects the nucleic acid. As you can see, surface बना रहा है, एक membrane, एक layer बना रहा है, and it enables the virus to attach to its host cell. तो यहाँ पे कुछ टेल्स होती हैं बैक्टीरिया की, which is not in part of your course. इसके थ्रू बैक्टीरिया आपका अटैच होता है ऑन इट्स होस्ट सेल, वो आपको स्टडी करने की जरूरत नहीं है। ऑल वायरसेस बाय डेफिनेशन आर पैरासिटिक, दे डिजीज कॉजन, दे सर्वाइव एंड मल्टीप्लाई विद इन द होस्ट सेल। एंड किस तरह करते हैं? बाय यूजिंग द होस्ट सेल प्रोटीन मशीनरी, बाय यूजिंग द प्रोटीन मशीनरी machine of host cell. Protein machinery is another fancy name for ribosome. Protein machinery, राइबोसोम ही बना रहा होता है प्रोटीन्स आपके। दिस इज अनदर फैंसी नेम फॉर योर राइबोसोम। इतना छोटा सा 15 मिनट्स का टॉपिक था आपका वायरस का, एंड इतना छोटा सा ही है पूरे कोर्स में। This was it for today's class. Tomorrow we'll be doing endosymbiotic theory and then we'll start the last section which is microscopy, and then we'll end this chapter. Monday को इंशाल्लाह we'll be doing past paper practice, जिसकी बुकलेट मैं आपको सेंड कर दूंगी आज थोड़ी सी देर में। एंड सैटरडे एंड संडे ऑफ है, तो हमारे चैप्टर 1 वीक में कंप्लीट हो गया, तो आई डोंट थिंक देयर इज एनी नीड फॉर एक्स्ट्रा क्लास। सैटरडे संडे आपका ऑफ है। Is this clear? Any difficulties, any issues?